For this PowerPoint, I would like you to be able to describe how deep brain stimulation of the subthalamic nucleus. I'm going to emphasize the subthalamic nucleus, in my explanation here. There are other reasons, like part of the substantia nigra that can be targeted here, but just for the sake of having to only explain one mechanism here, I am going to explain this through the context of the subthalamic nucleus. It's thought to treat tremors in Parkinson's disease. I would like you to be able to compare and contrast Parkinson's and Huntington's disease. I would like you to be able to identify distinct neural regions of the basal ganglia and cerebellum, and when covered in the PowerPoint, how they relate to specific behaviors. I would like you to describe tardive dyskinesia and the basis of it, the neural basis, and what it shares in common. And I would also like you to generally describe the extrapyramidal motor system. So we've already introduced the basal ganglia. It is one of the parts of the extrapyramidal motor system. It is distinct from the cerebellum, which is also part of the extrapyramidal motor system, and both interact with the accessory versus primary motor cortex in distinct ways. The basal ganglia sort of fine-tunes movement, as does the cerebellum, maybe in slightly different ways. The basal ganglia is thought to be very much involved with sort of the automation of behavior. We've already talked about this a little bit in the context of learning a habit, like learning a skill, a procedure, like learning how to play the piano. That would be represented here in the basal ganglia. Learning how to hit a golf ball, very much a basal ganglia refinement. And there does seem to be sort of like a shift from the more ventral portions to the more dorsal portions within the basal ganglia. The basal ganglia is thought to interact with the primary, the cortex in general through these sorts of loops that you can somewhat see alluded to here. The thalamus, as you might not be surprised, is a key sort of gatekeeper between the two here. Most of the information from the basal ganglia would be relayed through the thalamus after being split, really, between the external and internal segments of the globus pallidus, and working back one further to the dorsal striatum, back one further to the dopamine neurons in the midbrain. And this would ultimately then lead to activity in M1 of the primary motor cortex. So that's one big difference here. We've already learned about the primary motor cortex and how that sends down information through the corticospinal tract. The basal ganglia, by contrast, is the extrapyramidal motor system, which would sort of refine, allow for more refined movements that would then be directly conveyed down to the muscle. The basal ganglia are four brain nuclei that modulate movement. The caudate and the putamen are together known as the dorsal striatum. The substantia nigra is a nucleus that innervates this structure, the dorsal striatum, with dopamine. The dopamine neurons from the substantia nigra project to this region, and it's the degeneration of these dopamine neurons that leads to Parkinson's, whereas in Huntington's, you see more degeneration in the caudate and the putamen. Here's just a simple image from your book showing this anatomy. The caudate and the putamen are together in the striatum. In the rat, they have homologs called the dorsolateral, which would be the putamen, and the dorsomedial, which would be the homolog of the caudate. And across both species, these would be projected to by the substantia nigra. One difference between primate and rodent anatomy here is the primate, including the monkey as well, you can see clear separation between the caudate and putamen by something known as the internal capsule. This is missing in the rat, that clear segregation between the two brain structures. And here you can see another dissected human brain here, again from my neuroanatomy lab. All credit to Wake Forest School of Medicine. This is their work. But you can see in here just beautifully pigmented substantia nigra neurons. Notice the black pigmentation there. That's actually neuromelanin, similar to melanin that gives certain skin darker pigmentation. And this is really, it's fascinating to me that, I mean, these neurons just appear dark at this gross level. It's also worth pointing out that this level of this neuromelanin is much more pronounced in humans than non-human primates, and much more pronounced in non-human primates than lesser animals like rodents. It's thought to play some role in neuroprotection. Uh, and interestingly, it grows darker as we age. So, this would probably have been from an pretty aged adult, seeing how the neuromelanin is so well developed, where it would be much lighter in a child's brain. The primary disorders of the basal ganglia are Huntington's disease and Parkinson's disease. These would be both associated with an increase in the lateral ventricles here. Like most neurodegenerative diseases, they're associated with an increase in ventricle size. It's really due to a loss in neurons. It's not like the ventricles are just getting bigger. It's that you're losing neurons around the ventricles. But both you would see great degeneration in both Parkinson's and Huntington's disease. There are many proposed causes of Parkinson's disease. Again, I don't know that there's just one cause. There have been several gene mutations that have been suggested, including PINK1, DJ1, alpha-synuclein, Parkin, UCHL1, and LLRRK2. Many of these involve changes in mitochondrial function again. Looking at this as like one of the primary first dominoes, maybe across most neurodegenerative diseases. It takes about a loss of 80% of dopamine neurons for symptoms to manifest. And this is not purely only a genetic thing, although it depends on how you view genetics, I guess. Epigenetics, of course, is the interaction of the environment with your genes, and it could be the various environmental factors could turn on genes that lead to neurodegeneration based on experience, stress, virus exposure, or drug exposure. MPTP is one drug that it can cause Parkinsonian-like symptoms almost overnight. This was most pronounced during the 1980s, when a basement chemist in California tried to make a synthetic form of heroin known as MPP. Unfortunately, he didn't make MPP. He made a new drug that we didn't know about yet called MPTP. MPTP destroys the dopamine neurons of primates. Interestingly, it isn't as effective at destroying the dopamine neurons of rats. We have to use a different substance called 6-hydroxydopamine to lesion the dopamine neurons in the rats. But MPTP destroys the dopamine neurons of primates and humans. And when people in California started to take this drug in the 1980s, many of them woke up the next day unable to move, sort of like statues, overnight. Some of them received the very first, stem cell treatments for Parkinson's disease, these grafts. They didn't go so well. I mean, you'll find a PBS documentary saying that they did quite well. That, they're getting better. They're getting better with these sort of stem cell implants. But yeah, that first study didn't work out so well. And, this is a bit gossipy, but I can't help myself. One this is total speculation, absolutely not scientifically based, but might be interesting from a pop culture perspective. One sort of conspiracy theory is that, this MPTP could have touched Michael J. Fox's hands. It was around the same time that Michael J. Fox started to show symptoms of Parkinson's disease. I would be willing to bet that he was going to some parties where MPTP was. Certainly heroin was. Maybe he got a light partial dose. Who knows? Maybe he didn't. But there were other people on a movie set that he was working at in the 80s that also developed Parkinsonian-like symptoms. maybe it was nothing to do with MPTP and he did have early onset genetic-based Parkinson's disease. Either way, it's a travesty. It is, frankly, maybe it should speak a little bit about drug regulation. I mean, maybe it's better to legalize and have basement chemists making our drugs. But, that is for another conversation. But definitely fun to think about, right? Parkinson's disease causes tremors, loss of muscle tone, and difficulty in motor movements. Patients show degeneration of dopamine-containing cells in the substantia nigra, which project to the striatum. As previously alluded to, there are several genes that have been implicated in Parkinson's, but there's not like one set gene. One is a defective gene that encodes for alpha-synuclein, a basal ganglia protein that can be an inherited form of Parkinson's. Another heritable form of Parkinson's is a defect in a gene which encodes for parkin. Defects in either protein can cause the abnormal clumps called Lewy bodies, which are thought to promote neurodegeneration and alternatively could be a bit of an epiphenomenon. But regardless, they occur as neurodegeneration occurs through, at the very least, a correlation. L-DOPA is a precursor to dopamine, and its use reduces some symptoms. A lot of this work was done initially by a neuroscientist by the name of Arvid Carlsson, went on to win the Nobel Prize for his work, showing that dopamine has a role in movement. What he did is he took rabbits, and he used a drug called reserpine to deplete their vesicular stores of really all monoamines. And then he treated the animals with L-DOPA and showed that restoring dopamine alone was sufficient to render these, now paralyzed animals mobile again. And this went on to be characterized, in clinical trials by the famous movie Awakenings, which I highly recommend. It's a great classic from, I believe, the 80s. Electrical stimulation of the basal ganglia can also reduce symptoms of Parkinson's disease, but this requires deep brain stimulation and an implantable electrode. Neurotrophic factors and stem cells are also experimental treatments. In addition, there are things like, ultrasonic things being developed, things that are not invasive. Maybe even transcranial magnetic stimulation one day if we can get accurate and deep that far. There are issues with all these treatments. L-DOPA, one issue is that you can become tolerant to it, and then the super sensitivity that you get when it wears off and then you take a hit can lead to all sorts of problems. There have been case studies of people like just going on these gambling binges or sex binges or, all sorts of like odd behaviors when you're sort of coming on and off of L-DOPA therapy. Deep brain stimulation is a whole other can of worms. One, we don't completely know how it works, but there are some fascinating results out there that I have a TED Talk here from Lorenzen. This is the guy that really did a lot of the early work with deep brain stimulation, and he will tell you not only about how he has applied it in some of his historical case studies, but also how it might be used in other things like the treatment of depression. Deep brain stimulation is sort of the opposite of how it sounds. The last thing that you're doing is sort of stimulating release in the region. There are a few theories out there. One is that you're actually, hyperpolarizing everything by sending these sort of stimulations. So everything is sort of like in a hyperpolarized state. The other is that really over time you're lesioning the area, which would produce a similar effect to the hyperpolarization, either of which would be more sort of like an inhibition than a stimulation. But, the truth is we really don't completely know how deep brain stimulation works. And I highly recommend looking at this TED Talk. However, you can stop here if you're only interested about what's on the quiz. I'm not going to quiz you on anything from this particular TED Talk. It's just for your own enrichment. So you can stop here if you want, or keep going if you want to hear from one of the leading experts on deep brain stimulation.
One of the things I want to establish right from the start is that not all neurosurgeons wear cowboy boots. I just wanted you to know that. So I am indeed a neurosurgeon, and I follow a long tradition of neurosurgery, and I'm going to tell you about today is adjusting the dials in the circuits in the brain, being able to go anywhere in the brain and turning areas of the brain up or down to help our patients. So as I said, neurosurgery comes from a long tradition. It's been around for about 7,000 years. In Mesoamerica, there used to be neurosurgery, and there were these neurosurgeons that used to treat patients, and they were they knew that the brain was involved in neurological and psychiatric disease. They didn't know exactly what they were doing. Not much has changed, by the way. But they thought that if you had a neurologic or psychiatric disease, it must be because you are possessed by an evil spirit. So if you are possessed by an evil spirit causing neurologic or psychiatric problems, then the way to treat this is, of course, to make a hole in your skull and let the evil spirit escape. So this was the thinking back then. And these individuals made these holes. Sometimes the patients were a little bit reluctant to go through this because you can tell that the holes are made partially, and then I think there was some trepidation, and then they left very quickly, and it was only a partial hole. And we knew they survived these procedures. But this was common. There were some sites where 1% of all the skulls have these holes, and so you can see that neurologic and psychiatric disease is quite common, and it was also quite common about 7,000 years ago. Now, in the course of time, we've come to realize that different parts of the brain do different things. So there are areas of the brain that are dedicated to controlling your movement, or your vision, or your memory, or your appetite, and so on. And when things work well, then the nervous system works well, and everything functions. But once in a while, things don't go so well, and there's trouble in these circuits, and there are some rogue neurons that are misfiring and causing trouble, or sometimes they're underactive and they're not quite working as they should. Now, the manifestation of this depends on where in the brain these neurons are. So when these neurons are in the motor circuit, you get dysfunction in the movement system, and you get things like Parkinson's disease. When the malfunction is in a circuit that regulates your mood, you get things like depression. And when it is in a circuit that controls your memory and cognitive function, then you get things like Alzheimer's disease. So what we've been able to do is to pinpoint where these disturbances are in the brain, and we've been able to intervene within these circuits in the brain to either turn them up or turn them down. So this is very much like choosing the correct station on the radio dial. Once you choose the right station, whether it be jazz or opera, and in our case, whether it be movement or mood, we can put the dial there, and then we can use a second button to adjust the volume, to turn it up or turn it down. So what I'm going to tell you about is using the circuitry of the brain to implant electrodes and turning areas of the brain up and down to see if we can help our patients. And this is accomplished using this kind of device, and this is called deep brain stimulation. So what we're doing is placing these electrodes throughout the brain. These are, again, we are making holes in the skull about the size of a dime, putting an electrode in, and then this electrode is completely underneath the skin down to a pacemaker in the chest. And with a remote control, very much like a television remote control, we can adjust how much electricity we deliver to these areas of the brain. We can turn it up or down, on or off. Now, about 100,000 patients in the world have received deep brain stimulation. And I'm going to show you some examples of using deep brain stimulation to treat disorders of movement, disorders of mood, and disorders of cognition. So this looks something like this when it's in the brain. You see the electrode going through the skull into the brain and resting there. And we can place this really anywhere in the brain. I tell my friends that no neuron is safe from a neurosurgeon because we can really reach just about anywhere in the brain quite safely now. Now, the first example I'm going to show you is a patient with Parkinson's disease. And this lady has Parkinson's disease, and she has these electrodes in her brain. And I'm going to show you what she's like when the electrodes are turned off and she has her Parkinson's symptoms, and then we're going to turn it on. So this looks something like this. So the electrodes are turning off now, and you can see that she has tremor.
Not with the right hand side.
Okay.
I can't.
Can you try to touch my finger? That side's a little better.
That side is better, yeah.
We're now going to turn it on. It's on.
I just turned it on.
And this works like that, instantly. And the difference between shaking in this way and not The difference between shaking in this way and not is related to the misbehavior of 25,000 neurons in her subthalamic nucleus. So we now know how to find these troublemakers and tell them, gentlemen, that's enough, we want you to stop doing that, and we do that with electricity. So we use electricity to dictate how they fire, and we try to block their misbehavior using electricity. So in this case, we are suppressing the activity of abnormal neurons. We started using this technique in other problems, and I'm going to tell you about a fascinating problem that we encountered, a case of dystonia. So dystonia is a disorder affecting children. It's a genetic disorder, and it involves a twisting motion, and these children get progressively more and more twisting until they can't breathe, until they get sores, urinary infections, and they die. So back in 1997, I was asked to see this young boy, perfectly normal. He has this genetic form of dystonia. There are 8 children in the family. 5 of them have dystonia. So here he is. This boy is 9 years old, perfectly normal until the age 6, and then he started twisting his body. First the right foot, then the left foot, then the right arm, then the left arm, then the trunk, and then by the time he arrived, within the course of 1 or 2 years, the disease onset, he could no longer walk, he could no longer stand, he was crippled, and indeed the natural progression as this gets worse is for them to become progressively twisted, progressively disabled, and many of these children do not survive. So he is one of 5 kids. The only way he could get around was crawling on his belly like this. He did not respond to any drugs. We did not know what to do with this boy. We did not know what operation to do, where to go in the brain. But on the basis of our results in Parkinson's disease, we reasoned, why don't we try to suppress the same area in the brain that we suppress in Parkinson's disease, and let's see what happens. So here he was. We operated on him, hoping that he would get better. We did not know. So here he is now, back in Israel, where he lives, 3 months after the procedure, and here he is. On the basis of this result, this is now a procedure that's done throughout the world, and there have been hundreds of children that have been helped with this kind of surgery. This boy is now in university and leads quite a normal life. This has been one of the most satisfying cases that I've ever done in my entire career, to restore movement and walking to this kind of child. We realized that perhaps we could use this technology not only in circuits that control your movement, but also circuits that control other things. And the next thing we took on was circuits that control your mood. And we decided to take on depression. And the reason we took on depression is because it's so prevalent. And as you know, there are many treatments for depression with medications, with psychotherapy, even electroconvulsive therapy. But there are millions of people, and there are still 10 or 20% of patients with depression that do not respond. And it is these patients that we want to help. And let's see if we can use this technique to help these patients with depression. So the first thing we did was we compared what's different in the brain of someone with depression and someone who's normal. And what we did was PET scans to look at the blood flow of the brain. And what we noticed is that in patients with depression compared to normals, areas of the brain are shut down, and those are the areas in blue. So here you really have the blues, and the areas in blue are areas that are involved in motivation and drive and decision-making. And indeed, if you're severely depressed, as these patients were, those are impaired. You lack motivation and drive. The other thing we discovered was an area that was overactive, area 25, seen there in red. And area 25 is the sadness center of the brain. If I make any of you sad, for example, I make you remember the last time you saw your parent before they died or a friend before they died, this area of the brain lights up. It is the sadness center of the brain. And so patients with depression have hyperactivity. The area of the brain for sadness is on red hot. It's the thermostat is set at 100 degrees. And the other areas of the brain involved in drive and motivation are shut down. So we wondered, can we place electrodes in this area of sadness and see if we can turn down the thermostat? Can we turn down the activity? And what will be the consequence of that? So we went ahead and implanted electrodes in patients with depression. This is work done with my colleague Helen Mayberg from Emory. And we placed electrodes in area 25. And on the top scan, you see before the operation, area 25, the sadness area is red hot, and the frontal lobes are shut down in blue. And then after 3 months of continuous stimulation, 24 hours a day, or 6 months of continuous stimulation, we have a complete reversal of this. We're able to drive down area 25, down to a more normal level, and we're able to turn back online the frontal lobes of the brain. And indeed, we're seeing very striking results in these patients with severe depression. So now we are in clinical trials and are in phase 3 clinical trials, and this may become a new procedure if it's safe and we find that it's effective to treat patients with severe depression. I've shown you that we can use deep brain stimulation to treat the motor system in cases of Parkinson's disease and dystonia. I've shown you that we can use it to treat a mood circuit in cases of depression. Can we use deep brain stimulation to make you smarter? Anybody interested in that? Of course we can, right? So what we've decided to do is we're going to try to turbocharge the memory circuits in the brain. We're going to place electrodes within the circuits that regulate your memory and cognitive function to see if we can turn up their activity. Now, we're not going to do this in normal people. We're going to do this in people that have cognitive deficits. And we've chosen to treat patients with Alzheimer's disease who have cognitive and memory deficits. As you know, this is the main symptoms of early onset Alzheimer's disease. So we've placed electrodes within this circuit in an area of the brain called the fornix, which is the highway in and out of this memory circuit, with the idea to see if we can turn on this memory circuit and whether that can in turn help these patients with Alzheimer's disease. Now, it turns out that in Alzheimer's disease, there's a huge deficit in glucose utilization in the brain. The brain is a bit of a hog when it comes to using glucose. It uses 20% of all your, even though it only weighs 2%, it uses 10 times more glucose than it should based on its weight. 20% of all the glucose in your body is used by the brain. And as you go from being normal to having mild cognitive impairment, which is a precursor for Alzheimer's, all the way to Alzheimer's disease, then there are areas of the brain that stop using glucose. They shut down, they turn off. And indeed, what we see is that these areas in red around the outside ribbon of the brain are progressively getting more and more blue until they shut down completely. This is analogous to having a power failure in an area of the brain, a regional power failure. So the lights are out in parts of the brain in patients with Alzheimer's disease. And the question is, are the lights out forever, or can we turn the lights back on? Can we get those areas of the brain to use glucose once again? So this is what we did. We implanted electrodes in the fornix of patients with Alzheimer's disease. We turned it on, and we looked at what happens to glucose use in the brain. And indeed, at the top, you'll see before the surgery, the areas in blue are the areas that use less glucose than normal, predominantly the parietal and temporal lobes. These areas of the brain are shut down. The lights are out in these areas of the brain. We then put in the DBS electrodes, and we wait for a month or a year, and the areas in red represent the areas where we increase glucose utilization. And indeed, we are able to get these areas of the brain that were not using glucose to use glucose once again. So the message here is that in Alzheimer's disease, the lights are out, but there is someone home, and we're able to turn the power back on to these areas of the brain. And as we do so, we expect that their functions will return. So this is now in clinical trials. We are going to operate on 50 patients with early Alzheimer's disease to see whether this is safe and effective, whether we can improve their neurologic function. So the message I want to leave you with today is that indeed there are several circuits in the brain that are malfunctioning across various disease states. Whether we're talking about Parkinson's disease, depression, schizophrenia, Alzheimer's. We are now learning to understand what are the circuits, what are the areas of the brain that are responsible for the clinical signs and symptoms of those diseases. We can now reach those circuits. We can introduce electrodes within those circuits. We can graduate the activity of those circuits. We can turn them down if they are overactive, if they're causing trouble that is felt throughout the brain, or we can turn them up if they are underperforming. And in so doing, we think that we may be able to help the overall function of the brain. The implications of this, of course, is that we may be able to modify the symptoms of disease, but I haven't told you, but there's also some evidence that we might be able to help the repair of damaged areas of the brain using electricity, and this is something for the future to see if indeed we not only change the activity, but also some of the reparative functions of the brain can be harvested. So I envisage that we're going to see a great expansion of indications of this technique. We're going to see electrodes being placed for many disorders in the brain. One of the most exciting things about this is that indeed it involves multidisciplinary work. It involves the work of engineers, of imaging scientists, of basic scientists, of neurologists, psychiatrists, neurosurgeons. It's really at the interface of these multiple disciplines that there's the excitement. And I think that we will see that indeed we will be able to chase more of these evil spirits out from the brain as time goes on. And the consequence of that, of course, will be that we will be able to help many more patients. Thank you very much.